everyone. My name is Yawen Li from the University of Washington. Welcome to Astatine User Group Meeting 2021. It's a great honor to have 5 excellent panelists with us today. On the agenda, we have a short introduction first, then each panelist has 15 minutes to present. Then finally, there will be 35 minutes for Q&A. Please feel free to put your questions into chat. The panelists will answer them at the end. Okay, next. Although Astatine-211 is considered as one of the most promising alpha-emitting radionuclide for targeted alpha therapy, there are only a limited number of facilities in the world that can produce and work with this radionuclide. According to the recent review article by Dr. Yutian Feng and Dr. Michael Zalutsky, currently there are 5 sites in the United States, 2 sites in Europe, and 6 sites in Asia, with 5 facilities in Japan and 1 in China. Only a few of these facilities have produced the astatine at multi-gigabecquerel levels, although most of them can produce more than 1 gigabecquerel or 27 millicurie per batch. There is an ongoing effort to expand astatine production in the US, Europe, and Japan, either through the construction of new cyclotrons or by reviving existing facilities. An additional 9 facilities have been identified as potential astatine source in near future. 2 of them are in the US, 5 are in Europe, and the last 2 are in Asia. The most commonly used method for astatine production is to irradiate bismuth with alpha particles by the alpha-2N nuclear reaction. However, the short 7.2-hour half-life of astatine makes distribution of astatine beyond its production site difficult. The Radon-211 Astatine-211 generator system provides a potential solution for this problem. Radon-211 can be produced via the lithium-6 4N or the lithium-7 5N nuclear reactions. The half-life of Radon-211 is 14.6 hours. A Radon-211 Astatine-211 generator allows Astatine-211 to grow and reach its maximum activity at 14.5 hours. Over 80% of the maximum astatine activity will still be available after 24 hours. However, the production of Radon-211 and the development of the generator system are being investigated at only a few institutions and are in very early research stage. Nonetheless, it is a highly valuable method for improving the availability and distribution of astatine. To date, our clinical experience with astatine is limited to only 2 completed phase 1 trials and 2 ongoing phase 1, phase 2 trials. One of the completed clinical trial was conducted at Duke University for the treatment of recurrent brain tumor. In this study, astatine labeled anti-tenascin antibody was given locally in a resection cavity. Results from 18 patients treated with up to 347 megagigabecquerel or 9.4 millicuries showed no dose-limiting toxicity. Low-grade neurotoxicity was observed in a few patients, but no grade 3 or 4 toxicity was related to the treatment. The other completed clinical trial was conducted in Gothenburg, Sweden, for the treatment of ovarian cancer. In that study, a Fab2 antibody fragment targeting the sodium phosphate transport protein 2B was labeled with astatine, and 12 patients were treated intraperitoneally. The astatine dose level was escalated to 215 megabecquerel per liter, and no signs of radiation-related toxicity was observed. Moreover, the astatine therapy did not decrease tolerance to relapse therapy. More recently, the collaboration between the University of Washington and the Fred Hutch Cancer Research Center has enabled 2 ongoing phase 1, phase 2 clinical trials using the astatine-labeled anti-CD45 antibody conjugate BC8B10. This reagent is being evaluated as part of hematopoietic cell transplantation conditioning regimens for patients with leukemia or non-malignant diseases. A total of 43 patients have been treated as of last month. Recently, 2 new phase 1 clinical trials have been approved and are coming up for the treatment of multiple myeloma. I will talk more on the status of these Seattle-based clinical trials in a few minutes when Rob, Emery, and I give updates for our institution. As we might all agree, we are facing many challenges in realizing astatine-targeted alpha therapy. Production at the clinical level is still a challenge. We need more sites that are capable of producing clinical quantities for multi-center research trials. Currently, there is no commercial automatic system available for isolation of astatine after cyclotron production or for the synthesis of astatine radiopharmaceuticals. The development of such automatic systems can take years of effort. The labeling chemistry and distribution are also hurdles of astatine targeted alpha therapy, and ultimately, more astatine reagents need to be introduced into clinical trials to demonstrate the safety and effectiveness of astatine reagents. At the end of this introduction session, I would like to introduce our panelists and thank them for joining us. We have Professor Erica Prebys from University of California, Davis, Dr. Mehran Makvandi from University of Texas A&M, Professor Mehran Makvandi from University of Pennsylvania, Professor Michael Zalutsky from Duke University, Rob Emery, the director of the University of Washington Medical Cyclotron Facility, and myself. Without further ado, the first presentation is astatine production update for the University of Washington. This is a joint presentation. I will present the first half, then turn it over to Rob. The funding, the radionuclide production program at the University of Washington Medical Cyclotron Facility has been under the DOE IP stewardship since 2018. DOE-based funding partially supports the cyclotron operations. The radiochemistry program is funded through DOE base and R&D funding. We have been providing Astatine-211 through the NIDC since 2015, and we are developing methods for producing other radionuclides through R&D funding, which will also be provided through the NIDC when routine productions are achieved and facilities are adequate for high levels of activity. The amount of astatine produced and used at our site has more than doubled since our first clinical trial started in 2017. Over 80 gigabecquerel or 2.1 curies of astatine was produced per year in 2018 to 2020. 2.24 curies was produced in 2020, despite the challenges of the COVID-19 pandemic. We expect to be at a similar level or higher this year. 52 gigabecquerel or 1.41 curies has been produced in the first 6 months of 2021. In 2020, about 5% of the produced astatine was provided through the NIDC to astatine customers. 7% was used for research in automation of astatine isolation and development of quality control methods. 16% was used for preclinical biological studies to develop new astatine radioimmunotherapy reagents. The rest of the astatine produced was used for the 2 ongoing clinical trials. Here are some details on the activity shipped to our astatine customers. The batches we shipped so far were all 14 millicurie or 0.52 gigabecquerel at shipment. We can ship up to 50 millicurie or 1.85 gigabecquerel per batch. Unfortunately, due to the short half-life, there will be only about 10% of the shipped quantity after overnight shipment. The astatine is shipped in a near neutral solution in a plastic V-bottom vial. The typical volume is 0.6 to 0.7 milliliter, and it will always be less than 1 milliliter. FedEx overnight shipping service is typically used if the customer is within driving distance from Seattle. A local courier can be arranged for faster delivery. Our preclinical research using astatine has 2 primary goals. One of them is to improve the availability of astatine through the development of automatic astatine isolation systems, and more recently, the design of new astatine target station and new bismuth target. Rob will give an update on the progress in the target station and target in the second half of this presentation. The development of new astatine radioimmunotherapy reagents in animal models is achieved through collaborations with Drs. Sandmeyer, Green, Orozco, and Water at the Fred Hutch, and Dr. Park at UW. These preclinical programs are focused on new therapies for blood cancers, HIV, and hepatocellular carcinoma. Our first astatine clinical trial started, excuse me. Our first astatine clinical trial started in 2017. The PI is Dr. Brenda Sandmeyer. This clinical trial evaluates astatine-labeled anti-CD45 antibody in combination with fludarabine and total body irradiation as conditioning for hematopoietic cell transplantation for leukemia patients. Patients received escalating doses from 100 to 555 microcurie per kg, or 3.7 to 20.4 megabecquerel per kg of astatine BCA-B10. 33 patients have been treated as of last month. Dr. Sandmeyer presented the preliminary results of dose escalation at the transplantation and cellular therapy meetings in February. The second clinical trial using the same astatine reagent started in 2020 and has treated 10 patients so far. We are very excited that 2 new phase 1 clinical trials are coming up this year. These will use an astatine-labeled anti-CD38 antibody as part of conditioning regimens for stem cell transplant for patients with multiple myeloma. The clinical trial led by Dr. 211 will study astatine-labeled OKT10 and fludarabine alone, or in combination with cyclophosphamide and low-dose total body irradiation as conditioning before stem cell transplant. The clinical trial led by Dr. Damien Green will study the combination of astatine, OKT10B10, and chemotherapy drug melphalan as a conditioning regimen. With that, I thank you. I thank you for your attention, and a quick advertisement for our symposium on targeted alpha therapy at Pacific Chem in Hawaii this December. There will be a number of presentations on Astatine. With that, I will turn it over to Rob now.
Okay, great. So thank you. So yeah, I'm Rob Emery, and I'm the director of the cyclotron facility here at the University of Washington. And I'm just going to give you a little bit of an overview as what we're doing on the accelerator side and some of the challenges that we see in producing astatine. Just a quick overview of our facility. Accelerator came online in 1984 for fast neutron therapy. And as part of that original grant, it was specified that we could produce deuterons or a deuteron beam to replicate the work at the nuclear physics lab. And then because of that, we were able to then develop an alpha beam. And so that's what we're currently using for the astatine production, 29 MeV alpha beam. We do support other activities here, fast neutron therapy, obviously, isotope production. Again, with our alpha and deuteron beams, we can do things that other people maybe can't do. Our two biggest production items are Astatine-211 and Tin-117M. There are some other isotopes we're making currently for research purposes that Yawen's group deals with. We've also seen quite a bit of business lately from radiation effects testing, protons and neutrons. A lot of the space companies, SpaceX, Amazon, with their Kuiper project, putting up a lot of satellites and requiring more testing facilities. And this has been a nice new stream of income for us. And then finally, some proton therapy research. Flash, high dose rate, mini beam, spatially fractionated beams, RBE studies. So we're doing a lot of different things here that have to fit in with our isotope production. So the big challenge for us with the astatine is the alpha beam. It's much more destructive than our proton beam to our accelerator and our beamline transport system. You can see here on the left, our ion source chimney. We're using a cold cathode ion source. And the cavity here on the left is where we're producing our alpha beam from. And you can see it's just much more damaged. We get some metal spires coming off of that, which at times can short out the whole source, which takes us about an hour and a half to fix. And that can be a challenge if you're dealing with a short half-life isotope. You can see here some of the cathodes we use. The top one is a hafnium carbide, which we again use for the alpha beam, versus the lanthanum hexaboride, which we use for the proton beam. And these cathodes have about the same number of milliamp hours on them, but you can see that just the alpha beam is much more destructive, that cathode. And then over here on the right, we have our D-tip, one of our D-tips, which also acts as a source polar. And here's where we've made a modification to accommodate our alpha beam. You can see that we went from a solid D-tip to a separate removable piece, and then instead of going from a copper window, we had to go with tungsten, just because that copper window would get torn up with that alpha beam. So there are some definitely challenges working with this beam. Also with our beam transport, this alpha beam comes out much wider than our proton beam. So with the support of DOE, we've dropped in a higher power quad set there to focus the beam, more beam on target. We can easily come out of the machine with 100 microamps, but maybe you only get 60 of those onto target. And so this higher focusing quad will help us get more of that beam onto target. And then with this focusing, it's really important to know what our beam profile looks like, and how that's going to affect the energy deposition under the target. Our standard wire loop scanners won't work because, again, they get torn up by the alpha beam. And so we've gone with a new fiber scanner from a company, D-Pace, and it's worked quite well, and it's given us a very nice picture of our beam profiles on target. So that's been another upgrade we've put into place. And then sort of a final thing we're working on is target tree development. Up till now, all of our astatine production has been on a target designed and built at Triumph. And it's worked great. But a few years ago, working with DOE, we decided that it might be beneficial to come up with our own target and target tree based just on our experience and improvements we might make, and then something we could possibly share with the other producers in the DOE network. And so we've done that. And some of the requirements for the development were an automated ejection system, either to a remote transport system or a shielded container. And then we figured if we could do that, we could also probably do automatic target loading. And then the final sort of requirement was that it would work with a variety of target materials, we do a lot of different research isotopes, and so we wanted a single target station that could work with all of these. And then as far as the target itself goes, we wanted it to be able to withstand 100 microamps of our alpha beam. We're not quite there yet, but we hope someday to get to that current level. The target size itself needs to be compatible with a commercial remote retrieval system. This is something we plan on adding in the next year or two. Obviously low cost, easy to manufacture. We didn't want the target housing materials to interfere with any of the current chemical processing methods. And then finally, we wanted to minimize the target housing activity for safe handling because we don't have a remote retrieval system yet. Where we are in the development, so we've designed and manufactured the target and target station. We've done test runs up to 16.5 microamps and produced about 5 millicuries on the new target. And we've had successful testing of the automatic target loading system. Things left to do, I think in next week or the following week, we're gonna produce some more astatine for our radiochemistry group to isolate to make sure we're not introducing anything they don't want into the system. We need to finalize the manufacturing of the ejection system. We haven't finalized that because we're waiting on a couple of tools, but we were successful with the target loading system, and this part of it's going to be easier, so we don't see a problem there. And then finally, the last couple of things will be to increase our maximum alpha current, which is right now about 50 microamps, and then modify our other research targets to fit our new target station. And I apologize, I don't have pictures or videos of it. It's nice, it's pretty, but we're still working on the IP protection part of it, so then we can safely share it with our DOE partners. Just a quick overview of what it looks like to make astatine for a clinical trial run. We're making 130 millicuries at the end of beam. We'll come in after neutron therapy the day before and configure the accelerator. We get an operator in about midnight and bombard the target till about 7 in the morning. And then the rest of the day is slated for isolation, labeling, quality control. And then finally, we're ready for infusion in the late afternoon. And so you can see it's a bit of a tight schedule. And so if there are problems on the accelerator side, we have to make decisions whether or not we can get this to the patient on time, because there are a lot of people depending on this, or we need to abort early. And one of the things, small things we've come up with to help with this, but I think it's been quite helpful, is this just display on the operator control system that basically gives us an end of beam estimate based on our current target and the current activity level. And this does two things. First of all, if we go down for an hour or two for a problem, the operator knows how much activity is there or has decayed away and can make a judgment call on whether to continue with the run. Which is quite helpful. And then the other sort of issue is that with our limited staffing, I mean, this is the total, there's our total list of personnel that basically account for all activities at this accelerator site. And so if we have one person to run overnight, the next person to do neutron therapy, and then a few for support, it becomes sort of a tight schedule. And so we really have to sort of be lean and optimized, and small tools like this are quite helpful to the operator when they're producing. And yeah, that's all I have. Thank you.
Thanks for the opportunity to tell you what we're doing at Duke with astatine. It's pretty much what we've been doing for a long time, which has been very fortunate to take advantage of the facility that we have here for making astatine as well as other nuclides. I appreciate Yawen pointing out the recent review that Yu-Tian Feng and I put together and was published in Nuclear Medicine and Biology. We tried to capture what everybody was doing both in the US and around the world with astatine. Hopefully we got most of it right, but the important thing is, as Yawen pointed out, there are many more institutions that want to get involved in astatine, and some will definitely be on board within the next year or two. And those include the new machine that is being put in at Polatom, just outside of Warsaw, as well as the CS30 machine that has been resurrected and is going to be operational, I think, at the beginning of the next calendar year at Ionetics in Michigan. So The status for astatine production has been fairly constant at Duke for the last 10 years, which has been fortunate. The good thing that we take advantage of is an internal target that was designed by George Hendry, specifically for astatine production. Unlike everyone else, we've been running at about 28 MeV alpha particles as our incident energy. As I'll mention later on, we're going to try to join the crowd at 29 MeV because that will allow us to produce a lot more. We do about 1 production run per week. Sometimes we've done as many as 3. The maximum level of astatine that we've produced has been 9.3 gigabecquerels. However, our typical run, we run at about 48 particle microamps for about 3 hours, and that gives us about 6.8 gigabecquerels at the end of bombardment. And we've been doing these in support of some collaborative research we've been doing with Johns Hopkins, and this has allowed us to deliver more than 10 millicuries of astatine up there after having it be driven by courier up to Baltimore. So the CS30 is very reliable. We have not had too many problems other than the usual in keeping an old machine alive, but it is an old machine. And, you know, as we move forward towards addressing the needs, not only of investigators at Duke, but at other places in the United States, we need to make sure that this CS30 is as reliable as possible. So this is what the machine looks like now. A lot of this is going to be disappearing in the hopefully near future. Right now, with funding from DOE, we are updating all the subsystems in the cyclotron. John Vincent at Ionetics is the individual who has been in charge of this project. He's just done an incredible job in getting this done quickly and, you know, solving the usual problems that you have in trying to modernize a 35-year-old machine. So we've been dealing with some of the more physical subsystems, air compressor. We're working on addressing the cooling water issues. You know, we're in the South, and when it gets hot, we get condensation just about everywhere. That has just about gone away. We now have spare ion source power supply. They've designed a new rack for housing all the other power supplies. And that will allow us to consolidate many of the components and give us a much more smaller footprint for everything that we have. So there are some other updates that are still in progress. They have, John has designed a PLC-based control system to allow us to get into the 20th century or maybe even the 21st century. That has all sorts of readouts that will facilitate maintaining a stable beam. We've got a new knob meter. We've got a console power distribution panel so we can keep track of everything in one place. And this will allow us, as I said, to consolidate a lot of that mess that I showed you previously. Now, the next thing that we're going to do is once all of this is in place, we've not needed to go up to 29 MeV yet because we've been able to have sufficient activity for just about anything we've envisioned doing at Duke by running at 28, but with anticipated increased demands both at Duke and elsewhere, we will go up to 29 MeV. Before we do any production runs for other people, certainly in anything for stuff that will be leaving the cyclotron, we will evaluate radionuclidic purity. This will be done by the Triangle University's nuclear lab, which is actually located at Duke. It's nice to have an outside group that can do this. We're also evaluating alternative target geometries for our internal target. where we're going to try to refine the geometry a bit to make it easier to perhaps separate the bismuth layer from the aluminum backing plate. And one thing that we did, actually, it was part of that review that Yutian and I put together, is to try to figure out what kind of patient dose production levels you could do with a machine like the CS30 once we went up to 29 MeV. So as I mentioned, 3 hours at 28 MeV gave us, or routinely gives us 6.8 gigabecquerels. At 29, we should be able to get up to about 14. We looked at some of the papers that came out of the Gothenburg group, where they have a nice automated synthesis module and distillation module. We looked at the yields they got and the time that it took for the whole process. And then we figured, based on these yields and times, that we could get roughly 607 megabecquerels per gigabecquerel of astatine in the target. Based on our past experience and some of the things we're planning to do in the future, a typical patient dose would be about 370. So then a 3-hour run at 29 could produce 23 patient doses. So if you figure that if no one else is using our cyclotron and we were able to dedicate it to a patient study, let's say a multi-center patient study or a commercial product, if we did 2 runs a day, 5 days a week, 50 weeks a year, we could produce over 11,000 doses just on one cyclotron. And, you know, I know we've been a little charitable with some of these assumptions, but I don't think we're too far off what we could do in the future. So I think this bodes well compared with other alpha emitters that may be under consideration, for which supply is also an issue, but it's not an issue in the same way as it is for astatine. So quickly, let me just tell you about some of the projects that we have ongoing at Duke with the astatine. We're working on PSMA inhibitors. We're working on PARP inhibitors, but I won't talk about that today. Gold nanostars, VHH molecules, also known as nanobodies. And then we're also obviously working on next generation astatine labeling technologies, mostly for proteins, but also for other molecules. So for PSMA, we've gone through a very focused medicinal chemistry campaign in collaboration with the group at Johns Hopkins. And what we've been trying to do is to come up with a molecule that gives us good tumor uptake with relatively low kidney uptake. We also monitor stomach and thyroid, obviously, to look for in vivo stability. These are just some of the molecules that we've evaluated. And of these, this molecule called VK0290 with lutetium in the chelate looks the best. You can see good tumor uptake, very quick clearance from kidneys, and very little activity in the stomach. So this looks like a winner, and we expect to move this forward. And in fact, we have done studies therapeutically, both in the subcutaneous model and metastatic model. Looks very good. This has been accepted and should appear sometime fairly soon, I hope, in Journal of Nuclear Medicine. And we did not see any toxicities in a 13-month NTD study in CD1 normal mice given at therapeutic doses. So we intend to clinically translate a molecule very similar to this. We actually found one that's actually a little bit better that we expect to have in patients within 6 months. We're also looking at gold nanostars. These are like nanoparticles, but actually look like stars. This gives you a lot better surface area. This is a platform that was developed by Tuan Van Den and now worked on by Yawen Li, who's a postdoc in Tuan's group and my group. Very high labeling efficiency, very high stability in vitro. This is the biodistribution. Look down at the bottom, you'll see very low deastatination rate in vivo. Thyroid uptake and stomach uptake, these are percent per organ, is quite low. We've done a very preliminary therapy study intratumorally in a GBM, and we were able to show some benefit in terms of tumor growth delay. We're also looking at small proteins. They're called nanobodies or VHH molecules or single domain antibody fragments. This is what they really look like. They've got 3 different CDR regions. And we've been doing studies with one of our labeling methods with 5F7, which is an anti-HER2 VHH molecule developed by Ablinks in Ghent in Netherlands. And with a single dose, we have been able to get really, really good therapeutic response. In fact, I've seen similar studies with Actinium-225 and similar molecules, and we are actually doing much better with a single dose of astatine in this BT474 model. So I don't know when we'll get this into patients. Hopefully, we will also get this into patients sometime within the next year or so. Obviously, it takes a very, very good team to get all this work done. We have very good chemists in the radiopharmaceutical chemistry lab that have done all of the labeling, the cyclotron facility. We have 2 very dedicated operators, and Sean and Michael Daly. Ionetics has done a fantastic job updating our machine and helping us with future plans. And this work has been funded both by the DOE and the National Institute of Health. Thank you.
Thank you, Michael, for the great talk. Let's move on to the next speaker, Dr. Lauren McIntosh from the University of Texas A&M. Her presentation title is Steps Forward in Astatine-211 Production and Chemistry at Texas A&M University.
All right, so first things first, I just want to clarify, I'm from Texas A&M University. We don't have any association with the University of Texas, in case anyone is confused about where my association was on a couple of things for this user meeting. But I work here at the Cyclotron Institute with a great team, and we're developing the ability to produce and separate and hopefully one day provide Astatine-211 for the community. So I do want to make sure that I let you know what the takeaways are before I get started and into the weeds on everything. We've been working really hard on beam development, and I have been able to get a much higher current at a much more stable level over the past year, which is really exciting for us. We've made some good developments, and that's our team working in conjunction with all of our accelerator physicists and people here. So that's been really great. And we've been developing our targets so that they're thinner, there's less total bismuth that needs to be dissolved and then dealt with in the separation process. We have a graduate student, Laura McCann, who's been working with an ultrasonic soldering iron that's been very successful. And we're running it at 90 degrees from the beam and directly cooling the back. And between making the targets with this ultrasonic soldering iron and directly cooling the back, we have not melted any targets this calendar year. So if you remember the melted target pictures that Sherry showed last year, we haven't had as many of those lately, which has been really great. And we're working on our target extraction, monitoring everything. We're looking at, we have an air monitoring system in place to make sure that we're not having any volatilized astatine, because as those of you who have worked with it know, astatine can volatilize fairly easily, and we don't want any of that in our vault or anywhere else. And then we're making some strides in our dissolution and separation process. So we have showed that we can load a column with up to 50 millicuries of astatine, and the column can handle it, which is very, very exciting. So now I'll tell you about all the details on these things. So we're working with a K150 Cyclotron. So this was installed in 1967 and was decommissioned for a time while we used our other cyclotron at the Cyclotron Institute, and came back up not too long ago, but will partially for the purpose that we could have more intense beams and possibly be able to make some isotopes. So this is a zoomed in version of our floor plan here at Texas A&M. Here's a photo of the cyclotron, and we are working on the beam line that goes right behind this magnet. It's so small that it's not even on this drawing here. So we've got this chamber, and we've got here is that air handling monitoring system I was talking about, and our target goes right in the back here. And we're running at 28.8 MeV total and in a +1 charge state. And there it is. So I was talking about how we're making these bismuth targets. So here we have our graduate student. She's working with, this is Laura. She's working on the hot plate ceramic spatula, and this is that ultrasonic soldering iron, which actually uses acoustics to knock off some of the oxides on the aluminum. We get a much better adhesion of our bismuth onto the aluminum plate. So she's experimented with a few different things with the soldering iron, and also there's a weight here trying to press things out. And she has found that the soldering iron does the best job. One of the things that she'll be working on in the future is trying to, after the soldering iron does a really good job of getting the bismuth attached, trying to get rid of some of these ridges here, but they look great and they've been working really well for us. She's been going for about a 70 micron thickness, and we'll continue to try some things out. The target apparatus, what we have going on right now is our water is now directly cooled on our target. So we were running chilled water through a block that was behind a target. So the target and the block had to connect really well in order to have the heat transfer, and it was not as successful as we would like. So we're now directly cooling. So we've got water flowing in through the center and then out on the outside here. So this is our aluminum frame for our target, and this is where the bismuth goes. Currently, we're using a flat target, but we have used a well in the past. And then this is us trying to test our targets before we put them in beam. We've got an industrial heat gun here and our target with our little thermocouple to try to see what's going on with the temperature. Cooling. This is before we were able to get beam for the year, but this system also works rather well. We've also been interested in our beam uniformity. So our method to try to tackle that right now is we've got this this 3D printed structure that's on an actuator arm that we can put into our beam and then pull back out in front of our target if, when we need to. So this isn't a real-time thing, but something that we can look at while we're tuning or when we want to. So we've got 3D printed, there are 37 brass screws, which are all connected to a wire. So the current is read off of each of them. And these currents go into this nice little LabVIEW program where we can actually visualize, okay, this area is hotter. So if we move it down, we can spread it out some more. We can see how much beam we have approximately from a relative standpoint on our target. We don't put our whole beam on this 3D printed thing, obviously, but we can see sort of what the spread looks like. And then we've got this surface is covered with the phosphorescent cadmium sulfide, so we can visualize, we can see it here and we can see it there and know that that corresponds well. So that's been good. This is looking at, we're also reading the current off of our target. So this is our looking at that later. And so we were seeing for a while that we would get some beam and then it would kind of die off. We come in and peak it and then it would die off. So we were watching this happen overnight. And so this is from December of last year. And we were able to give some real time, okay, this is what's happening to our beam overnight. When whoever tunes it is going home, what can we do about that? And we were able to do something about that. So this is looking from last month, just a couple of weeks ago. We have much more beam on target and it's much more stable. We do have some times where we'll have the deflector will spark, or we'll have, there are electronics that are involved in our interlock system that are having a little bit of trouble with the amount of neutrons we've got in our vault. So we're working on that. But the big takeaway here is that we're much more stable. And that has led to us being able to create more astatine, which is very exciting for us. We're also looking at the target area with the cadmium telluride, where we can get the characteristic X-rays from Astatine-211. And this is helping us to confirm a clean extraction, which is something that our rad safety encouraged us to do. We're working on correlating this to the amount of astatine that's actually produced. And so that'll be a nice check that we'll be able to look at. So hopefully eventually look at our current, what we would expect to be produced, and then the cadmium telluride before we even pull the target out to tell us what we're making. I alluded to this or showed this to you in place a little bit earlier. This is our air monitoring system. So we've got this here connects to the chamber, and then we've got this connects down to a pump. So we're pulling the air from the chamber, and this is inside, pulling the air from the chamber down and around. There's a filter here, and we've actually added, there's another filter here. So these are just the fiberglass filters that will catch astatine, we hope, right? So we've seen it work before, and they'll catch the astatine, and we'll be able to see those characteristic alphas in the silicon detector before we even open the chamber up to air and expose anyone in the vault to any sort of astatine in the air. This year, when we haven't had any targets melting, we haven't seen any astatine on these filters. So this check is working, and we're producing astatine without getting it all over the air inside of our chamber. And so this is just a quick summary of what we've been able to make. I want to draw attention to down here in July, we did a couple of irradiations that made over 50 millicuries. So that was a huge leap forward for us, very exciting. So the different colors just mean this was the old target when we were at 10 degrees with the chilled block. The red is the chilled block at an 80-degree angle from the target, from the beam as we were moving towards this 90-degree target. And then the green is our current configuration, where we've got a 90-degree target. It's a coin target that is directly water cooled. So we're excited about that. On the separations, column chemistry side of things, after we produce the astatine, we take the target to the radiochemistry lab and dissolve the target in nitric acid. And so then this solution, our bismuth and astatine solution, goes through this column, which is an amber chrome resin, which is impregnated with 3-octanone, which will interact with our astatine in a way that you can see in this paper. It'll get more into it if you're really interested, and that will hold on to the astatine and allow us to wash off the bismuth and then elute off the astatine later. So here you can see the fractions. So we've got the loading process, and we're getting a lot of our bismuth 207 off. And then we have our wash here. And then when we're stripping, we're getting basically just astatine, and we're able to get most of it off. All of these are counted with our high purity germanium detector. So this is just our characteristic Astatine-211 spectrum. And we've moved from some larger columns to some micro columns, and have even been able to send a column successfully to some collaborators at MD Anderson Cancer Center. So they were able to do chemistry with it, which is a really exciting leap forward for us. We're also continuing to study liquid-liquid astatine extraction, because the fundamental chemistry of astatine is not very well understood, due in part to the fact that all of astatine is radioactive and it's not easy to get your hands on some. So the radiochemists here at Texas A&M have been looking into this. So this is looking at extraction in 1-octanol as the organic solvent. And we're able to fit this data and understand more about the interaction of the astatine with the 1-octanol eventually, we hope. So this is just, this is what we call our dissolution box here. And we've got our target down in the bottom, and then there's nitric acid, where some of the brown is just the NOx gas that you're getting off of that. So that is also an ongoing area of study. So there's the extraction, and then looking at if you reduce or oxidize, what difference that makes in that liquid extraction. So that is another area of active research right now. So the main takeaway points are that we have approximately doubled our astatine production capability in the past year. The column extracts the astatine from the bismuth very quickly, and we are in the process of working on an automated system for this. And we have shipped via local courier to MD Anderson, which is exciting for the future as far as being able to have people who know a lot about radio labeling and can use Astatine-211 to great effect. We're also looking into some fundamental chemistry, and we hope in the future to be able to make Astatine-211 available through the NIDC. So this is our Astatine-211 team here at Texas A&M. And I do want to mention our operations folks, both our radiation safety. Dan Menchaca has been fantastic working with us to make sure we're doing things safely and well. And Brian Roeder has done a really great job of developing our alpha beam and knowing what it is that we need to do in order to get us more of it. So I also need to acknowledge other folks at A&M, because you can't run a program like this with just our core team, as well as all of our sources of funding, which it takes a lot to run a facility and beams like this. So I will be happy to take your questions, I believe, after everyone's done doing their presentations. So.
Yes, we will hold the questions to the end. Thank you, Laura, for a fantastic presentation. Very impressive progress in astatine production. And I apologize for mistaking your institution. Thank you for correcting that. We are actually right on time. And so let's move on to the next speaker, Dr. Eric Prebys from the University of California at Davis. The title of his presentation is Development of Astatine-211 Production at UC Davis.
Okay, so I'm going to be talking about the development of Astatine-211 at UC Davis. Our team, I'll start by introducing the team. I'm the PI, but my background is accelerator physics and high energy physics, so this is kind of new to me. Dan Siebra is the nuclear physicist involved. We've got Michael Backfish is the person in operation who runs the beam studies and basically does a lot of the work on developing this. Brian Devine is our head machinist who designed and fabricated the target. And we have two graduate students. One is Kathleen Stewart. She's worked on the chemistry end of it mostly to develop the astatine processing, and Lena Korkaila is a new grad student who's been working mostly on cyclotron modeling and helping with operations. A little bit of orientation. UC Davis, we refer to it as centrally isolated. It's a sleepy little college town, but you know, in an hour or two, you can be skiing, you can be in San Francisco, you can go to wineries, whatever you want. It's also conveniently located about 20 minutes from the Sacramento airport. But more importantly, you know, here in this area, there are lots of people who would be potentially interested in Astatine-211, and the nearest source of it is University of Washington. So we feel we've talked to people and there's a need for it. Just a little bit about the cyclotron. It's got a very interesting history, which I can't really go into in detail because of the time. The yoke of this cyclotron, the magnetic yoke and the central pole pieces, were actually part of Ernest Lawrence's 60-inch cyclotron that was built in 1939, and it's actually where Astatine-211 was discovered, as well as plutonium. In the 60s, they decommissioned this to build the 88-inch cyclotron, and Lawrence's former student, John Youngerman, was at Davis, so he convinced them to send the steel. But the design was completely redone based on the design of the Oak Ridge isochronous cyclotron at the time. And it was commissioned in 1966. You know, how did we decide to get into this game? Well, for several decades, the lab has actually been self-supporting through charging to provide beam for UC San Francisco's radiative oncology team for 1 week a month to treat uveal cancer. So we have a medical treatment line. Our beam energy is too low to do proton therapy on deep tumors, but it's sort of it's perfect for uveal cancer. So this is a very successful program that's been going on since I believe 1994. The rest of the time we sell beam to study radiation effects on materials and electronics. for a diverse group of users. We have some 50 or 60 different users, primarily in the aerospace industry. And through that, the lab basically supports itself. We're similar to like the K150 at Texas A&M, but we're much less subscribed. We run 5 days a week, 8 hours a day, usually. A few years ago, the university determined that simply providing a service for a fee was not really part of the university mission. So they decided to either expand the mission of the lab or shut it down. So it was separated from something called the Air Quality Research Center, because long ago it was used to do environmental studies, to which it had been attached. And I was brought in 4 years ago with a mandate to increase the scientific output of the facility. And that it's that general. To that end, I can do whatever I think is appropriate. We determined, it was actually Dan Sebra's suggestion, that developing Astatine-211 production might be a good use for the cyclotron. It's well matched to capabilities. Of course, we'll be using the same reaction that you just heard about at Texas A&M, alpha beam on a bismuth target to produce Astatine-211. And again, as I said, there's a community of interested users with no nearby source. So some of the challenges, we knew there would be challenges. I'll show a layout in a minute, but Crocker Lab used to produce isotopes. It used to produce isotopes for the medical school and the vet med school. The last time it produced an isotope was 1994. So most of our users don't require high currents. So in fact, we could not deliver high currents, particularly in alpha particles, with our existing filament source. Our extraction efficiency for alpha particles is quite low. So we early on determined that an external target was really not the right way to go. So we'll use an internal target very similar to Duke's. There has never been a proper computer model of the cyclotron. It was built as a copy of the Auric cyclotron, and they basically got it working 50 years ago and haven't worried about it much since. And of course, like everybody else, we had a big unforeseen challenge come in. So the proposed technique is to use an internal target, but unlike the Duke University cyclotron, we can't accommodate both a probe and a target. So our plan or what we have done, is to build a hybrid probe and target. So here's our cyclotron vault. Here's our external lines, our radiation effects line, our therapy line, a neutron line. This is the historical isotope production line, which we have retasked to be a higher radiation area. People who want very high radiation compared to the usual aerospace users. This is our probe shown here. And luckily, the probe goes in through a little airlock. So we can remove this probe or target probe without breaking vacuum. And that's what makes this possible. So the scope of the project, our original scope was design and fabricate the target, outfit a lab here at Crocker in space that was vacated by the Air Quality Research Center. And we were going to investigate both wet and dry chemistry. We wanted to achieve 100 microamps of, that should be alpha beam on the target. And we estimated we could recover up to 60 millicuries in an 8-hour shift, where we had time for setup, 5 hours of exposure, and 2 hours for processing. Of course, like everybody else, we didn't get nearly the funding we requested. So we came up with a reduced scope that we would do the work in Professor Sebra's lab in the physics building, which is right next door to us, investigate only the wet chemistry. And our aim is to recover a few millicuries to establish the capability during this period. And then we'll see if people are interested. We would then work to increase our output. When I signed up for this panel, I really thought we would have accomplished this by this point. And we almost did. And I'll explain why we actually haven't done that yet. So our key progress and milestones, we actually now have a proper model of the cyclotron. Lena Korkaila did most of that work based on the Opal cyclotron model. And it was used to design the target orientation curvature. We put in a new filament supply that allows us to go. We were with our old supply, we were never able to exceed 1 microamp. We were mostly limited to about half microamp. Once we did the new supply, we very quickly hit 30 microamps, which is enough for our goals. Basically, once we put a new filament supply, it popped right up to 30 microamps. We did an alpha profile measurement in the cyclotron. We put some Gaffchromic film. We were somewhat surprised at the size of the beam. We knew it was about 1 centimeter vertically. we don't understand why it's so wide or horizontally. We're trying to understand that through the model, what's leading to that effect. It does explain why our extraction efficiency is so extremely low for alpha particles. But this is actually good news in that it makes it easier to distribute the beam and avoid sputtering or melting the target. We've also outfitted the lab, and we're all set up to do the recovery chemistry once we start producing astatine. And I don't show a picture, but we've also identified a high purity germanium detector to do the initial assays of the target. That's in our building. So how did COVID-19 affect us? Well, on March 15th, basically, we had a shelter-in-place order. Our lab never completely shut down. We continued eye therapy with extreme precautions, so 1 week a month. Basically, except for the simulation work, all process on Astatine-211 effectively stopped. We were given permission for limited operations in May, strict social distancing protocols. People were still encouraged to work from home. Priority was given to our users. The Astatine program slowly came back to life, but it was severely impacted. Basically, we returned to operation, but we were impacted by this time lost. So on to the target probe design. This is a conceptual design of the target. Basically, we have a leading and trailing beam probe, and we monitor the current on both of those and the target body itself. We're designing this for up to 100 microamps of operation. That's what the cooling is designed for. This shows the fabricated target. Basically, the target does not, I should have put a side view. The target is angled such that when it's mounted to the cooling channel, it has a correct angle. We don't have an angular adjustment on the initial target. We've also, I was looking for a good picture, and I unfortunately couldn't find it. We've also perfected the technique for coating this with bismuth. It turned out just painting it on with a steel wool brush will nicely put 100 microns or so of bismuth on the target. We did a series of beam tests in early March in which we monitored the current on all three probes, and we found that most of the beam was hitting the leading probe with very little current on the body or trailing edge. We modified the trailing edge to extend further, and we determined that our target angle was off by about a ¼ inch at the trailing edge. In other words, we built the target with the incorrect angle. When we went back and looked, this was essentially a misunderstanding in our modeling about the location of the target relative to the magnetic field. So we designed a new target and fabricated it, did subsequent beam tests, and we found we got much more uniform illumination between the leading and trailing edge of the body, and we should be able to get at least half the beam on the target, which is good enough to meet our initial goal. So our plan, our next step is to do a live test with this target, which we had intended to do a couple weeks ago. So we had planned to do we were going to do a first production exposure about a week ago, but I think I mentioned, it was amusing to hear Michael say that their machine is old because our machine is much older. So we had a pump fail a couple of weeks ago that brought the machine down exactly during the period when we had planned for our first run. So as soon as we get an opportunity, we are ready to go with our first exposure. Our first exposure, we're simply going to hit the target, verify that with a fairly low current, verify that we can hit it without melting the astatine, and then assay it with our high purity germanium detector. And then in the next exposure, we'll build our way up to a millicurie of astatine. So our time scale is within the next few weeks, which is of course past our funding period, but we've asked for a no-cost extension to complete our goals before the end of the year. So at that point, then we will see how much interest there is and whether it's practical for us to proceed with full production here. We're very interested in going that route. So I would like to acknowledge Ethan Balkin and the DOE University Isotope Network for funding this work, Scott Wilbur and his team at University of Washington for ongoing advice and for showing me their facility, and they've given us a lot of advice on the wet chemistry, Michael Zalutsky and his team at Duke for the same, for letting us crib off their internal to target design, and of course, pretty much everything I know about astatine is from reading his review papers. So thank you.
you, Dr. Prebys, for your excellent presentation. It is very interesting to hear about what's going on at your institution. We look forward to hear more as it continues. Let's now move on to our final presentation. From Dr. Mehran Makvandi from the University of Pennsylvania, and the title of his presentation is Astatine-211 Production for Translational Science in Radiotherapy.
Hi, thank you so much for that great introduction. And I really like to thank all the speakers today. I've seen a lot of really kind of creative ways to problem solving, and it's something that not everybody in the field of nuclear medicine is faced with, but we're lucky enough to study astatine, so we're faced with it daily. So today I'm going to talk a little bit about astatine-211 production and what we're doing at the University of Pennsylvania. So we recently re-outfitted a cGMP chemistry suite in our cyclotron facility for the manufacturing of astatine-211 radiopharmaceuticals for clinical trials. And I don't, if anyone's seen my presentations in the past, there used to be this big blue hot cell from Brookhaven, which looked like it was probably 30 years old or so. And so we've really invested into building a program that can span the gamut of really going from bench to bedside. But in order to actually get to the clinic, we needed to make a few modifications on our cyclotron system. And one of the biggest, or one of the key features to update on the JSW BC3015 cyclotron that we have was to actually just build a new target design. Similar to Texas A&M, we started out using a cold finger, and then we very quickly, with the help of Dr. Paul Ellison from the University of Wisconsin, realized that direct water cooling is the way to go. And ultimately, we had this target system designed and fabricated by Sumitomo Heavy Industries in a collaboration that we have ongoing, which is capable of handling electrical currents up to 20 microamps. And here's an image of it actually mounted onto our JSW cyclotron, and we still have 2 external target port stations that we're hoping to have targets for to perform actual other R&D test irradiations using coin target systems. And so our capabilities for production have really increased and even provided the opportunity for going into other areas of isotope production. At the University of Pennsylvania, we've traditionally separated astatine by dry distillation, and last year we presented a induction heater for actually doing the distillation, and this works very well. It's rapid. You could get nearly 70% of the astatine in like the first 5 minutes of the process, going from completely cool to hot, and then it cools just as rapidly. So my student, Paul Martirano, is really spearheading this, and he's doing a great job making new designs. However, after seeing the publication by John Burns and Sherry Yanello, I became very interested in looking at the chromatography extraction method. And I'm pleased to say that after attempting this actually 3 times, we were able to reproduce the results on the 3rd time and got excellent extraction recovery. And I'm sure with a little bit of optimization and just general comfort for handling nitric acid, we could get the time down as well, which is great. But I think last year when I gave this talk, I ended on a subject that I was getting into. And so the remainder of my talk today is really going to be focusing on what we did in the last year to study the biostability of astatine-211 radiopharmaceuticals. And I pose this question: if supply is able to meet demands, then what? So we saw all of these wonderful talks earlier, and it sounds like, we're going to even have a commercial supplier potentially by January 1st of next year. We have at least 5 or 6 academic institutions that are really revving up the ability to supply astatine. So like, what are the next big questions that we as a field have to answer? And I think the one thing that people always kind of throw at me when I talk about Astatine-211 for alpha therapy is there's a general uncertainty around the biostability of Astatine-211 radiopharmaceuticals. And it should be no surprise, astatine, like everything, like other halogens, has its own chemical properties that make it either unstable under certain biological conditions. And so the work we did over the last year was really aimed at trying to figure out, is astatine really all that different from another halogen? And so if you start by reviewing the work by Wilbur, and this even goes back further, but I was too young, so I could only understand up to 2004. So we'll start here. And what Scott did was really amazing, where he said, you know, I'm just gonna radiolabel some benzimides and compare astatine to iodine. And sure enough, the astatinated benzimides ended up with uptake in the thyroid or the neck, and the iodine did not. And so from this, a lot of the field, I think in general, started to suspect astatine was probably very different from iodine, and that it almost seems like people think that it cannot be stable in vivo. And so this is a concern I'm constantly faced with. And so I think something that I would like is to kind of reset everybody's mindsets about that exact topic. So flash forward to 2017, some work by Montavon and colleagues out of the group in Nantes in France, they performed quantum mechanical calculations using spin orbit density functional theory to actually estimate what the bond dissociation energy of a carbon astatine bond is versus carbon iodine. And the significance of this kind of technological breakthrough is astatine is such a large element. The inner electrons are moving nearly at the speed of light. So in order to understand how the quantum mechanical calculations will actually work, you have to go into this higher level of theory. And what they found is astatine indeed has a lower bond dissociation energy after oxidation, which means it's more susceptible to oxidative dehalogenation. And they proved this empirically. They showed under Fenton conditions, which can commonly occur in lysosomes, that hydrogen peroxide plus ferric chloride at these concentrations can deastatinate a statobenzoate over time. And so this kind of brought two things to the forefront of my mind is, in the first being, can we actually use these computational methods to predict bond stability and really find ways to define what would be a stable construct versus unstable construct? And secondly, is this actually an accurate scenario that would recapitulate what actually happens in, let's say, a patient? And so if we follow the work by Montavon a little bit further, he proposed a mechanism that has actually been proposed previously for iodinated antibody constructs that are internalized. And so it's very funny that we like to always say astatine is unique from iodine, and then we kind of take all of the history of iodine and apply it to astatine. And in the story, it kind of goes like this, where we have a targeting vector, which is an antibody or peptide. There's some type of linker moiety, and we have astatine. And when this radioimmunoconjugate actually binds to a protein complexes and is endocytosed, and then enters the lysosomal protein degradation pathway, it's exposed to a highly oxidative environment. And under this condition, our targeting vector is going to be degraded because it's made of amino acids along with the protein complex. And so we need to understand what's the fate of our radionuclide in the linker conjugate. And so for many astatobenzene constructs, if it's a carbon astatine bond, we know that under these conditions, the carbon astatine bond can be cleaved. And what happens at that point is it can exit the cell and redistribute and end up in the thyroid. And so our hypothesis that we would have to really accept our null hypothesis that carbon astatine bonds are unstable. However, there are two very nice examples in literature where there are stable astatine bonds. And one is Astatine-211 carboranes that was really pioneered by Dr. Wilbur. And the other is Astatine-211 benzyl guanidine derivatives, which was pioneered by Dr. Zalutsky. And so when we start to really think about what defines the in vivo stability of statinated radiopharmaceuticals, we can simply just start to evaluate our two known examples. Carboranes utilize a boron astatine bond rather than a carbon astatine bond, and it's actually thermodynamically more stable. Whereas benzyl guanidine really piqued my interest because it utilizes a carbon astatine bond. However, it's stable in vivo. So this really led us to wonder, what is the guanidine function or the benzyl guanidine substituent doing to the aromatic ring system to make astatine stable towards oxidative deastatination? And so we took a very elementary approach to kind of answer this question. And we looked at both electron withdrawing groups like an amide versus electron donating groups like a alcohol on an aryl astatide molecule. And what you can see in these molecular electrostatic potential maps is that the electron withdrawing groups are really pulling electron charge out to where the substituent actually is, and they're pulling this electron density away from astatine. So the phenol or the alcohol, which is electron donating, is actually increasing the electron density into the ring system. And we believe this is actually one of the nice ways to illustrate how halogen might be more susceptible to oxygen attack. So we had computational methods that were already suggesting, yes, the substituents do affect the susceptibility to oxidative dehalogenation. And this should be no surprise because it is pretty much the same for every other halogen that has ever been studied. So we went into empirical studies using both Fenton conditions and sodium permanganate, where we would expose these constructs to an oxidant and then measure stability over time. And to our surprise, we actually found a very interesting result. For astatobenzimide, it was stable enough to where it only decomposed about 75% over the course of 3 hours. However, the iodobenzimide was completely stable at 100 micromolars of sodium permanganate. And so this kind of led us to believe, okay, we can accept carbon astatine bonds are less stable than carbon iodine bonds. And this has been proven before by the work by Montavon. You know, the same goes for a carbon bromine bond versus carbon iodine. So everything's kind of following the trend. And what we found for astatophenol was actually very interesting. It decomposes extremely fast compared to iodophonol under these oxidizing conditions. However, when comparing iodophonol to astatobenzimide, the iodine agent's actually less stable. So this is a nice example just to show that, you know, there's no rule saying that all carbon astatine bonds are going to be less stable than a carbon iodine bond. It's actually going to be very circumstantial and dependent on the electronics of the system. And we in this work, we performed a series of experiments using aryl iodides with different R groups, and we found a nice linear correlation between the first ionization potential that's calculated by the SODFT method with the product stability. And so here you can see the alcohol is one of the least stable, and it has a lower threshold for the first ionization potential. And the nitro or the amide are some of the most deactivating or stabilizing groups. And so this kind of brought us to this point where, okay, we understand what the substituent effects are for aryl astatides, and it's very similar to what we saw previously with aryl iodides and other aryl halides. And so we wanted to look a little bit further and really start to understand, okay, there's got to be something else that leads to in vivo instability. And so thinking about what's the other side of the coin, enzymatic metabolism is going to be probably just as important for small molecules, if not more important for small molecules, which a lot of these original assumptions were based off of, including the astatobenzamides that Dr. Wilbur looked at. And sure enough, when we actually look at metabolic stability in a liver microsome assay, we find that the iodophonol is fully metabolized, but in the para position doesn't actually deiodinate. So even though it's more susceptible to oxidative dehalogenation, it's not susceptible to metabolic dehalogenation. And in contrast, one of the most stable deactivating groups, like the nitro, actually makes it more susceptible to a nucleophilic attack by glutathione. So there's an exact metabolic mechanism that would result in deastatination. So we're leaning more towards the substituent effects are really going to affect stability under oxidative conditions, and this follows the same properties for the electronics of the ring system. But there's also this other half of the equation, or potentially even more, and it's going to be the metabolic effects for dehalogenation. And so with this, we would like to kind of have this general hypothesis that not only do you have to be stable under lysosomal oxidative conditions, but also enzymatic conditions. And if we look at the known constructs that are stable, we have the astatocarburanes, which have a very high bond dissociation energy between the boron and astatine, greater than 60 kilocalories per mole. Even after oxidation, it remains very high. And then looking at meta-astatobenzylguanidine, we find some very interesting properties by calculating the first ionization potential, which is 249 kcal per mole. And the bond dissociation energy is for astatine, it's actually relative to what it is for iodine on a iodobenzoate. And even after oxidation, there's really not a big drop in the bond dissociation energy, which means this is probably stable as well. And so in contrast, we know statobenzoates are unstable under oxidative conditions, and here are some numbers to really help everyone compare what type of stability we're talking about. And so we know that these constructs would be susceptible to in vivo deastatination. We know these are going to be stable. And we even know that there's an example for the para-substituted benzylguanidine analog, which is it's more metabolically active. And so if you calculate the total energy for removing astatine from this molecule, you actually find that it's more stable than meta or ortho benzylguanidine. However, in vivo, this construct is actually unstable. And so, and it's known that in the para position, it's more likely to get metabolized. And so I think what to take away from this is that we're really building a framework to think about how the biostability of Astatine-211 radiopharmaceuticals works, so that we can explain why we're actually using our approach versus another. And so with that, I would just like to end with a brief update on what we have on the horizon. And so we're hoping to submit 2 INDs for astatinated radiopharmaceuticals by 2021. And those clinical trials will hopefully be on the way. And so with that, I would just like to thank my research group. Tara Tagby did all of the computational work. Paul developed the induction heater. Jonathan ran the metabolite assay. And I wanted to include a lot of pictures because I know it's been a long time since we've been able to really socialize. And so all of our past collaborators and hopefully we'll be able to have new collaborations in the future, as well as all of our funding support in the DOE Isotope Program. So with that, I'd be happy to answer any questions.
Thank you for the fantastic talk. The in vivo stability of astatine labeled biomolecules or any alpha emitting radionuclide labeled biomolecules is definitely maybe the most important question to answer. And it's very innovative to use computational methods to provide insights and guide designs of the labeling reagents. With that, we have completed all the talks for today. And if you have, for the audience, if you have any question for our panelists, please put them in chat or in Q&A. I see there are 2 questions in there earlier that have been answered by Rob Emery. One question is, are your currents particle or electrical? And the second question is 1+ or 2+ beam. Rob, would you like to comment more on those 2 questions?
Sure, I think I answered them there, but it's electrical current and 2+ alphas, yeah.
Thank you. We have another question from Eric Dorman to Mehran. Are you still using bismuth oxide targets?
So this was a project that we kind of had to put on hold temporarily. We still have plans to go through and do the irradiations and just put the data out there. But currently we're using the metallic bismuth as our standard route for producing.
Okay. Thank you. We will give it a minute to see if more questions show up in chat or in Q&A. So from Joe Kumlin, is anyone planning phase 2 or phase 3 clinical trials in the near future? Not sure. I will give it a try to this question. The ongoing clinical trials are actually phase 1 and the phase 2 at UW and the Fred Hutch Cancer Research Center. They do have the phase 2 component. We currently don't have any plans for phase 3. We think more advanced trials will probably need companies to be involved, and we definitely look forward to opportunities like that, but no real solid plans yet at UW. Anyone else wants to comment on that question? Okay, and another question from Eric Dorman. Lauren, you mentioned you have 28.8 MeV beam. Is that mirrored?
Yeah, so that is, I believe, based upon the frequency of the cyclotron. It's measured by the RF, so it's not externally measured with like a detector or something like that, but it's based on the RF from the cyclotron.
And Eric has another question. And do you see any 1181 keV gammas in your HPGE spectrum? I believe this is to Lauren as well.
I think we have. I don't know if that's something we see every single time. I want to ask Eric why he wants to know about that.
I think maybe that gamma ray comes from Astatine-211. Is that
yeah? So I think we've seen that before, but I don't have a quantitative answer for that. Yeah. Okay, thanks, Eric.
So Eric said yes, Astatine-211.
Yeah.
Yeah. Okay.
So yeah, maybe it's not like a noticeable, it's not a peak compared to the Astatine-211 though. So we're not super worried about it.
Yeah, it's still a non-settled question, like what level of polonium-210 can be acceptable. So yeah, it'd be a good discussion for later. So there is another question from the audience. I have a question for University of Washington. Can you easily provide this to the Europe or Middle East? Because the half-life is 7.2 hours. The answer is no. We have a hard time even providing it to customers on the East Coast. We really don't think it'd be too much decade away during transition if we ship anywhere further. Okay, next question is, can Texas A&M comment on the success of the chemistry performed at MD Anderson after Astatine-211 shipment? That's an interesting question.
That is an interesting question. So this has only happened this summer so far, so we're hoping we'll be able to show some stuff from that next year, but they were able to do some preliminary stuff. So hopefully we'll be able to comment on that next year.
Yeah, more to come. Yeah. For UC Davis, the support for the bismuth is aluminum or stainless steel. So what is the material for the target backing? Eric, could you please comment?
It's aluminum. The target itself is aluminum, and then the cooling body is copper.
I was actually curious about the color of the targets the target you showed. Our bismuth target, it has a silvery, white color. Yours look more golden, I think.
It might have just been that was just the target. It didn't have any bismuth on it.
Oh, I see. I see.
Yeah, but like I said, I have a picture somewhere, but I couldn't realize at the last minute I couldn't find it. But yeah, it looks like everybody else's bismuth target.
Okay, thank you. Koshin asked, how do all the universities manage the Astatine-211 production schedule? Especially UW-MC looks very tight to dedicate for Astatine-211 production. Does this already determine at least prior to a half year? Yeah, we schedule Astatine runs at least a week. I think usually at least a week ahead of time. I will let Rob comment on that more.
Yeah, so that's true. We like to get at least a week notice for the longer overnight clinical trial runs. It's nice to be able to be flexible for these trial runs because we'll find that patients come on and off the protocol pretty quickly. And because some of these protocols are sort of the last option for these patients, it's important that we're flexible enough to be able to produce when they need it. So sort of being nimble on that is important, yeah.
Thank you, Rob. Are there any attempts to start introducing metrology or is it too early? Dosimetry? I don't know who wants to tackle this one. Michael, could you say something about it?
Gee, thanks. So the dosimetry in patients and preclinical models, both cellular and animal, is a very active area of research. There are probably 2 or 3 places that have focused intensely on that, both in Europe and the United States. I think this is a very important area mostly for preclinical, because being that alpha particles have such an important subcellular distribution component, it's hard to envision how you would extrapolate that to actual patient dosimetry. So at least in my mind, the more esoteric dosimetry approaches where things are done on smaller dimensions is probably going to have its greatest impact on preclinical research. But of course, that will be important for informing clinical research.
Thank you, Michael. I agree. Dosimetry for alpha emitters is a very important area. So we have a question from Johnny Orozco for UC Davis. What kind of additional processing handling was required at UCSF before dosing into patients? Or did all of that need to happen on site before shipping out?
Maybe I wasn't clear. We have not actually shipped any astatine. Our cancer treatment is proton therapy. The UCSF oncology group that's a proton therapy line.
Do you envision that UC Davis will handle the isolation and radio labeling part? You know, in the future.
Yes, that is the plan.
Great.
We have a lab space here that we're beginning to outfit, or we'll do our initial processing in one of the physics labs, but we have a space here that we're outfitting to do whatever chemistry is required.
Understand. I hope this answers Johnny's question. 2 more questions are in one is general question to the speakers using wet chemistry instead of dry distillation. What are your predominant reasons for this? I will comment first. At our institution, there are several reasons. The first one is our radiation safety is really not comfortable with us distilling potentially volatilized radioactivity, especially alpha emitters. Yeah, that's the first reason. And the second reason is our bismuth target is bigger than the normal commercial coin targets for the production of other radionuclides. And it's difficult to fit the target into the commercial tube furnace. We would have to build a special device for dry distillation, and that has created some hurdles for us. I will ask Lauren to also give a comment.
It's similar reasons as far as radiation safety. Would rather we weren't doing this dry distillation. There was a group at Texas A&M before our current iteration of isotope production who wanted to try the dry distillation, and they did not have very good efficiency with the dry distillation process. And we have had a lot better time working with the liquid extraction than what we've seen published as far as the efficiency of the dry distillation. And it's pretty quick also, which is important with this short half-life.
Thank you, Lauren. Mehran, would you like to also comment on this question? Because you mentioned your group switched from dry distillation to the wet chemistry method.
Yeah. So we still do both. We haven't exactly switched, but I'm always interested in trying new things. And I saw the method that came out of Texas A&M. It looked very appealing to me. So we have a very small coin target. It's about the size of a nickel, and we have like 150 milligrams of bismuth on it. So when I was imagining dissolving, my bismuth layer in nitric acid, I get to use less than 1 milliliter of nitric acid. All of a sudden, everything doesn't sound so bad compared to, trying to dissolve something in a liter, or I'm not sure exactly, but how much you might need. And so thinking about it from that point of view and where the chemistry automation is going to go, we're interested in learning about that. And then also just doing chemistry with the astatine after it comes out of nitric acid extraction was interesting to us. But for right now, I think between our induction heater, we get much better reproducibility and efficiency compared to what we got with the tube furnace. And I think it's just because you're directly heating the coin target. And so you're releasing the astatine in a much more uniform manner. And we've also miniaturized our distillation tube it's like a cup this big now. And so the astatine can't stick to everything and it goes straight to your solvent trap. And so for us, it's really doesn't really matter. Whatever is going to work the best, we'll probably do that. And so I think there's pros and cons to both. And I think both are viable. The wet distillation or the wet extraction method has definitely had, I would say, a lot more advancement over the last, 10 years compared to dry distillation. Really not much has changed about it. It's still the same process. So I think there's more room for growth with the wet extraction.
Okay, great. Thank you for sharing your thoughts. I hope this discussion is helpful for people who are trying to decide which way they want to go. And let's move on to the
Actually, I just wanted to comment, I'll just add, we did it for this, we're going to go with wet chemistry for the same reason that our radiation safety people like it, even though I like dry chemistry because I understand that. So, but we're going to do the same thing, at least initially.
Thank you, Eric. If no more comments from the panelists, I will move on to the next question. To the general audience, what level of importance is the chemical purity of astatine products? To the general audience, I will give it a try. So we have with our boron cage labeling reagent, we've been getting a very consistent labeling yields, and we are monitoring the chemical purity of our astatine product, and the chemical purity is very high. The bismuth residue, bismuth impurity is definitely in the low micromolar level. We haven't found that to interfere with our reaction. So I will now turn this to the other panelists to let them comment, because I think this has to do with which labeling method you are using. Sorry, Michael.
Well, we primarily have been using tin precursors, and for patient use or anything that would be envisioned for future patient use, you obviously want to make sure that you exclude tin. And so we try to keep it at parts per million levels. And, we just do assays periodically to make sure that's where we are. Otherwise, I would say that the issues are basically the same as the issues for any radiopharmaceutical preparation intended for patient use. You want to have the purity very, very high. But in our case, it's not really totally the chemical purity you want to worry about. It's the radionuclidic purity. And there has been some mention about assaying for Astatine-211. That's absolutely critical. And this is one of the reasons why, frankly, we have stayed at 28 MeV, because we know there's no Astatine-211 at that incident energy. However, we will certainly, as I mentioned, investigate higher energies, and hopefully the radionuclidic purity will be acceptable.
Thank you, Michael. Yeah, I definitely agree. Radiochemical purity and radioisotopic purity are as important, if not more important, maybe than the chemical purity of Astatine products. Okay, let's move on to the next question. Lauren, can you please describe the Faraday cup again? I think I know you. I think I know you how you were using it. In particular, beam current while reading. Read time, do you turn the beam on and off again to make sure the 3D printed doesn't melt, or just low constant current?
So I think you're talking about the segmented Faraday cup that I show for our looking at our beam profile. So we just keep that at a really low current. We don't put microamps of beam on that. It's more like picoamps. We'll have some picoamps or maybe nanoamps. It's a very low amount of current that we put on there, so that we're not worried about melting the plastic, because obviously we're not worried about cooling that or anything like that. So it's on an actuator, and we'll have it in while we're tuning the beam and then pull it back out. So I think that answers your question, or how I understand your question. So, okay. Okay.
Yeah. Joe, comment again. Thank you. My apologies for the poorly written question. No, you're fine. It's quickly typing your question in there. Definitely, I think we understood what you mean. What QA does UW do before shipping Astatine? I can answer that question. We have HPGE assay that is conducted quarterly to validate we don't have any Astatine-211 in the Astatine produced. That is one of the QA tests we do. And for every batch, every run, we look at the radiochemical purity, where we determine the percentage of acetate versus acetate. We also do ICP-MS retrospectively to look at trace metal impurities in the Astatine solution. What else? I think that's all that we do. For Lauren, when you increase the current on the Faraday cup, it's quite possible the beam distribution changes. Have you done any validation between low current on probe and actual beam on target?
So we have not put like a radiochromic film in or anything to make sure that it's exactly the shape that we think it is based on the Faraday cup. We have noticed, so you can get some weird little scorch marks on your target while you're irradiating it, and those tend to be in a similar pattern to what we'll see on our Faraday cup. The beam halo looks pretty similar. So we haven't tested that extensively, but we haven't seen anything either that makes us think that the beam shape is vastly different when we're looking at a larger intensity. So.
Thank you, Lauren. Okay, that has been a great discussion. If no more questions, we will thank our great panelists again for their fantastic presentations, and for all of the audience for your time and attention. And we will see you again next year.